Welcome to Tech and Drugs, where we explore how AI is reshaping pharma and biotech. Today, we are joined by Rick Schneider, co-founder and CEO of Helical, a Luxembourg-based startup pioneering AI-driven drug discovery. Rick and his team are on a mission to democratize biofoundation models, making AI tools accessible to pharma and biotech companies. In just a short time, Helical secured $2.2 million in funding, backed by investors from AI leaders like Cohere and Hugging Face, to build open source platform for genomic and mRNA research. One of their major breakthroughs, training a cutting-edge mRNA model using Luxembourg MeluXina supercomputer, competing with models from top pharma companies. By combining AI with high-performance computing, Helical is accelerating drug development and pushing the boundaries of biotech innovation. Rick's vision goes beyond tech. He's building a collaborative AI-driven biotech ecosystem, partnering with leading universities and open source communities. So how will AI revolutionize drug discovery? Let's dive in. So Rick, welcome to the show.
Hello, Thibault. Thank you for having me.
Great. So before we go any further, can you tell me what's the name of the computer? Because I wasn't sure if it's MeluXina. Did I pronounce it right?
You got it right. It's called MeluXina. It's actually an HPC based in Luxembourg. And so we have a partnership with them in place since the model training for Helix. We can talk about this later, but Helix was quite a large model in that sense for training needs. And so this partnership helped us to pre-train this large model.
We'll definitely talk about that in a moment, but let's start from the beginning. So you had an interesting journey. Can you tell us, what led you to what you're doing today?
Yeah, happy to do that. I'm actually an engineer by training, generic engineering, no bioengineering. And I quickly transitioned into the world of data science and AI, worked at big tech companies, joined actually also a German scale-up, was called Celonis, and doing many different things. I worked in product, machine learning, go-to-market, and spent time in different cities. And that's really what I enjoyed doing, like this multiple things at a time, this entrepreneurial part. And for the backstory and maybe how I got into this field, actually, I always wanted to become a doctor, and I was signed up to become a doctor in medical school in Munich. And then last minute, literally last minute, I changed to an engineering class. Never regretted it, but yeah, so I was always very close to that field. And together with my co-founders, Maciej, who is a medical doctor and a genomics researcher, and Maxim, who is an AI researcher, yeah, we looked into this field of the intersection of AI and pharma, and got really excited by those new types of models coming out, those biofoundation models, and so decided to kick off Helical from there.
You're actually the second engineer that we have on the show that started really as a pure engineer and then transitioned to pharma. So, but as you said, I mean, there is, you had some hesitation earlier in your life, between life science and engineering. So what made you decide initially to go to engineering, and why then did you decide to focus your engineering skills on life science? So what was the motivation for both things?
Yeah, I thought as an engineer, I could anyways come back into this field. And I was always thinking of creating a company. And so I thought that the engineering skills would maybe help me more in that. And I was also undecided. And so this engineering route, I thought gives me like the broad approach to that. And I actually thought about then for my master's studying medical engineering, which I switched out for data science and AI. But yeah, again, a similar story here. So yeah, I was hesitant, but never regretted it.
So, it's interesting because lately I've been talking to a lot of pharma that are working more and more with engineers and tech companies. And they keep talking about people. So some people call them cyborgs, other bilingual or multicultural. So essentially, these are people that understand tech and understand life science. And that's a hot commodity at the moment. So you're a hot commodity.
I wouldn't say so, but thank you. But no, our entire team is this actually. Like Helical is really the intersection of those two worlds. We need super strong AI engineers to get all of that done. And we have really good biologists, and they are all either coming from the one side and super interesting in the other, or are coming from the AI side and really interested in learning a lot about the bio side. And I think that's a bit our superpower, to be honest.
Yeah, we'll definitely talk about that. I think that's a theme that I keep hearing. And I think this is also something interesting because a few years ago, there was not a real bridge between those two disciplines. And I still think that now that it's quite rare to have a curriculum that integrate both aspects. And looking forward, we'll definitely need more people like that. But let's start at the beginning. So when we had our first discussion, we talked about foundation models. And this is something I'm so interested in, and I think it's fascinating. But can you start by explaining what's a foundation model for someone who's not necessarily a techie?
Yeah. So foundation models are quite a paradigm shift in AI, and I will explain why. But a foundation model, to make it very simple, is basically a very large pre-trained AI model. Pre-trained meaning that it is being trained once, and then you can use it for multiple things, that has learned on very, very large amounts of unlabeled data. Unlabeled is key, and I explain what unlabeled means, but it uses a technique called self-supervised learning to achieve this paradigm shift. And it differs quite a lot from the more traditional AI algorithms, where the AI algorithm actually needed that labeled data to predict something very specific. Take an example. I don't know. For example, let's say you want an AI algorithm that predicts if a review is positive or negative, right? A review that a customer leaves for a restaurant. Where with traditional AI, what you would need to do is like create a list of reviews, and then a human person would need to read them all and say, hey, this review was positive or negative. That's called labeling. And then the algorithm would learn to predict if it is positive or negative based on this data set. Well, foundation models change this. They don't need this labeled data set. They can simply learn from reading tons of text, in the example of OpenAI, ChatGPT, for example. And then by just reading so much text, well, it all of a sudden develops the capability to also say if something is positive or negative, and hence say if a review is positive or negative. And that is a huge paradigm shift, right? Because you don't need this labeled data anymore. And if you pull that forward to the field of which we are in, so we are not in the domain of text, we're in the domain of bio, we are in the field of bio foundation models. And here things get really interesting because you use similar concepts from the traditional foundation models, but you learn on biological sequencing data. So raw DNA sequencing data, RNA sequencing data, can be single cell expression data, and so on. I don't go into too much detail here. But they learn to understand and read these sequences, so the language of life to a certain degree, start understanding it from the raw data, and then can be used for very specific use cases because they now have learned this underlying complexity in this data. And then using them on those use cases is really what makes them foundation model, because they can do so many different things, basically. They are the foundational brick, basically, on what you build for the AI models.
So I think at the moment the most famous foundation model in life science is certainly AlphaFold. I mean, it's the one that has a Nobel Prize. But AlphaFold solve one specific problem. So essentially, you have a protein sequence, and then you can predict the three-dimensional structure of a protein. So obviously, AlphaFold alone cannot solve drug discovery and development. So can you give a few examples of foundation models in biology, and what are the use cases they address, not only in biology, I mean in drug discovery and development?
Yeah. I think it can be argued if AlphaFold is a foundation model because of what you just said. It's very good at something very specific, but that's a different story. I think a more recent example of a true foundation model would come from Arc Institute. So Arc Institute is by the Collison brothers, very, very large budgets, and they have released Evo2 recently. Evo2 is trained on DNA and RNA sequences, a lot of them from multiple species. And it's actually very funny because in the training of this model, only one human reference genome has been used. So they only see one human reference genome, and yet by the sheer size of the model and by how many other DNA sequences it has seen, it predicts mutations better than any specialized model, state-of-the-art model that is currently out there. So just to give you an idea of what a foundation model would be. And Evo is really versatilely usable across all, let's say, DNA and RNA use cases you could potentially think about. And also, just one more remark here, Evo is a 40 billion parameter model. For those who don't know what it means it's very large. It's very, very, very large. It's actually the largest foundation model ever released open source, period. Not only in bio, fully open source. And that is crazy. And it shows how we are moving to an industry where this is happening in bio. And very precisely, some use cases we are currently addressing with our customers, they really go across the entire drug discovery stage, from things such as target identification, so using those models in very low data environment to find better targets, biomarker discovery so that you can stratify your patients for the next clinical trial, knowing the gene signatures that are responsible for success or failure. So those are the type of use cases you can address. And why it works so well is because those models have seen millions of sets, and the data sets that are being used by pharma are often very small. They maybe have like 50 samples, right, from a clinical trial. So how do you get something useful out of that? Well, foundation models are a great key to that.
So you're okay. I don't think that we will have time to discuss whether AlphaFold is a true foundation model or not. But what you said is interesting because, so do we need one very large foundation model or several? Because I'm always thinking, like we have so many types of data in life science. So you talked about sequence, but of course there is images. There are all the pathology images, for instance, and there are so many other things. So many other measurement that could be included, and that represent something happening, essentially a biological process. So how would it work? Because let me try to see how I articulate the question. There are so many questions you can ask in drug discovery and development. So as you said, it can start with, there is a disease, so I want to understand how what is the molecular foundation of a disease. What is the targets or the targets? Then how can I perturbate the target? So this is where we will say, okay, what happens if I block it? How do I block it? With what? Mm-hmm. Then the thing that I develop to block the target, whether it's a large molecule, a biologic, or a small molecule, it needs to work in a specific organism. You need to define, whether it will be toxic or not, and then how it will translate in human and in a larger population. So there are literally thousands of questions. So yeah, I'd love to understand, can one model solve everything? Is there, one to rule them all, or do we need to have several?
I honestly love that question because it's really spot on. And we have been thinking a lot about this. And our clear answer is there's not one model solves it all for many reasons. You need multiple modalities, right? You don't solve problems on one modality, but you could still argue that you can train across modalities over time, right? DNA, RNA, single cell, bulk, what do I know, images, you named it, right? So no, there won't be one model fits it all. We actually have a benchmarking framework where for each use case that we do, we look at all the existing models that are out there before we choose one. And funnily enough, it is often a different one coming on top that is very good at a bit more specifically your use case that you're targeting. I think the end goal here is to go towards a virtual cell where you leverage all of those different foundation models across those different modalities. And you start combining the so-called embedding spaces. This is basically the output of the foundation model into one digital cell where you can do all of the things you just mentioned. You can leverage it to do those perturbations that you have in mind. You can simulate it to be a diseased cell, and you can see in silico how you could heal that cell, basically. And this is all possible with foundation models. And actually, we take this approach at Helical by saying, look, we are model agnostic. We don't care which model you want to use, but we focus heavily on how you use the model and how you combine multiple models into one use case. So we are really the toolkit sitting between the models and the use cases, and we bridge that gap, basically. And to answer as many use cases and questions as you possibly could. And basically all the ones you just mentioned are part of what we solve with our platform.
So you mentioned virtual cells. I've seen a few papers in the last few months talking about virtual cells. How far are we from something actually really useful in real life applications?
So I think from the virtual cell itself, we are still a bit away, right? Because it's also about how you combine all of those things. But specific tasks this virtual cell could execute, we are getting closer and closer. Perturbation analysis is one example where we see a lot of progress, especially with recent perturbation data sets being released in open source. If you use them in a clever way with existing foundation models, and you do so-called fine-tuning on those data sets, so making the model specialize in this specific task, you actually get quite good results. So I think on the task level, we are getting closer, but as a whole system level, we are not there yet, where you would simply say, hey, that is now my virtual cell. I can do all of that things in one basically virtual cell. There we are not fully there yet.
So moving on, you're working with pharma clients. So what's the biggest hurdle, working with a pharma company, especially when it comes to them adopting AI, working with scientists who are not necessarily fluent in tech? So what's your experience so far, and how do you solve some of the potential challenges?
Yeah, interesting. I think the biggest hurdle is really, honestly, speed, meaning that to get from the moment where you decide, hey, there's an exciting use case we're going to work on together until you actually do it, there's a lot of time passing in between those. So there's this still a bit, slowness in big pharma sometimes. Then I think there's the topic of data, right? Data is the biggest asset for pharma companies. So under no circumstance, this data can leave their premises. So architecturally speaking, we need to adjust to that. We need to always be capable to either run on premise or on the private cloud or on whatever basically setup we're currently looking at with the customer. And then you mentioned the other one is like the AI nativeness. I think we often work with computational biologists that know a lot of traditional methods. So there is still, and more than fair enough, a certain skepticism against, hey, can those methods actually outperform the state of the art? So how we react to this is really, in all the benchmarks we do, we always include the state of the art methods, that more traditional methods, into the evaluations as well, because we need to prove still that AI models can reach those levels.
Okay, and obviously, data is the most important in our business. So how do you deal with poor data quality or legacy data? Because I worked with pharma for many years, and I always find it funny when people say, "Oh, pharma companies are sitting on a gold mine of data." Like, well, we sit on a lot of data, but not everything is usable. A lot of the legacy data is not labeled, is quite dirty, requires quite a lot of work actually to make it useful. A lot of the data was produced when, no one knew about AI. So obviously it was not prepared to be consumed by AI models. So how do you deal with that?
Yeah, that's interesting. Very interesting question. I think on many cases, the data sets are actually not that large that they can produce. So I think that is one of the key challenges, even though on an aggregated basis, pharma companies have a lot of data. Use case specific on the drug discovery program that is currently running, often the data sets are super limited, simply due to the fact that this data needs to be produced on the go, right, in the program. And often there's either only budget for, let's say, generating 10, 50 samples, or it's just simply too early to go to double down on those things. So I think the size of data sets is a huge issue. Then the other thing is quality. I think there's something in those sequencing experiments, there's something called batch effect, right? So if those sequencing has been done at different times in different experiments, while they will be deviating. Well, one of the promises of those foundation models is if you have seen enough cells across different batches and so on, well, they can start reading through the noise, which is one of the benefits you have, but still you need corrective measures to adjust those type of things. I think those are the main two hurdles that we see and that are key of bridging. And I think those are the biggest problems that pharma companies are trying to address on their drug discovery process. So I'm talking really about the earlier stage drug discovery use cases.
So you talked about a few technical aspects of working with large pharma. Have you ever encountered more cultural aspects or cultural blockers?
Yeah, I think this is really also what I try to refer to with let's say the traditional role of the computational biologist versus us trying to be completely AI first. Probably the reality should sit still in between those two chairs. But yeah, culturally, you are still very much focused in, hey, I need to benchmark those results. I need to understand that exactly what you're doing. I need to fully trust the predictions that you're doing. And it makes sense, right? Because in the end, the whole assets that they're going to develop based on those predictions, well, that's a lot of money is going to flow into that, right? So you have a lot of reluctance to a certain degree to trust results, and rightfully so. But that is, I guess, one of the biggest barriers you have.
So we talked about large pharma. Now let's talk about startup. So what was the inspiration for you and your co-founder for starting Helical?
Yeah. As I mentioned, I really wanted to become a doctor. The same is actually true for Maxim. Both we chose doing engineering. But I mean, we know each other since high school, and after university, we got together. Everybody worked a bit in his field. Maxim in AI research, Mathieu actually genomics research and cardiology, myself in big tech. And then we got together and really talked about, hey, how could we do something in the space of precision medicine? That was like the big question. We were like, surely there's opportunity with AI to be better in precision medicine. So we started looking at what models are actually out there, and we fell on one of the pioneers in the DNA foundation model space called nucleotide transformer from InstaDeep. And we started using this model, and while using it, we were like, okay, the results are pretty good where we can generate with this, but it's so hard to use it. There's such a gap in between, hey, here's the model, and here's what we want to achieve. And that's how, when we started thinking about, okay, it should not be that hard to leverage those models. It should not be that incredibly complicated to think of which use cases can I now use it to? How do I fine tune it? How do I bring the lab in the loop? How do I do reinforcement learning? How do I benchmark against the others? All of those questions popped up in our mind, and we said, okay, if we can solve them, and we come from the AI side, then pharma companies must be stuck as well. So we talked to pharma, found out that we were right with our assumption, and started working with them. That is a bit how Helical got started, basically.
Yeah, so that's an interesting point. How do you ensure that your solution align with the large pharma real needs?
I mean, you need to simply constantly talk to your customers, right? In the end, we came with some assumptions. We were very lucky with our first customer being very open to testing things with us. We were obviously very scrappy, 3-man startup at that time, and they gave tons of input. So we were capable of understanding, okay, what are the challenges? And they are not purely technical, even though this is how we first thought about it. They are also really like, what is the value you can generate with that? How can you move the process forward? How can you reduce wet lab, the need for wet lab testing? How can you improve the hypothesis that you realize? And by understanding those things, we iterate towards something that I think now has product market fit and is a really, really good platform that is used by our customers.
You mentioned before lab in the loop, so it's definitely an approach that a lot of companies are talking about. Really combining. You start in silico, you generate some hypothesis, then you go to the lab, generate the data, and then fit that back into the model. So do you already have now your own labs, or is the lab in the loop part of how you work with clients?
Yeah, so we don't have our own lab, and not planning to have so in the short-term future. What we do is we equip our customers with the technology to do that, meaning all the AI part from bringing the lab in the loop and improving models with feedback that you generate based on the predictions we did. It's called reinforcement learning, and there are many other techniques. We take care of that, basically. So we make it very, very easy for our customer to get into a lab in the loop type of setup by bringing back feedback we generate directly in the model and really making it their own model. Even though they started from something open, they added their data, they validated data, they generated lab data, and then they bring it back into the model to consistently improve predictions we are doing. So we are more facilitating the lab in the loop than basically doing it ourselves. And this fits as well our strategy, generally speaking, of saying, hey, we are model agnostic. We work with all type of models that are out there. Sometimes opportunistically, we develop one, right? It's what we did with Helix. It's a messenger RNA model. There's actually, at the moment, I don't know what's happening, but when we released it, it was popular, but now it's really getting super popular. It has now, I think, 10,000 downloads only last month, 30,000 in total. So something is happening in the community, but that's great. So yeah, we are really agnostic there.
So obviously, it's a very dynamic field at the moment. There was a very interesting database that lists all the tech bios or let's say tech companies that have applications and use case in life science. And last time I checked, there was almost 400 companies. So how do you position and differentiate yourself in this very competitive field?
Yeah, I think very importantly, we really focus on being the platform for the models. Again, meaning making it super easy for pharma to leverage those models in their very specific use cases. What we are not trying to do is leverage the models, for example, to advance our own pipeline, right? So we are not, in that sense, a tech bio company, but we equip the tech bio companies of tomorrow to be the most efficient at building AI and to get the best results. Because what we see as well, it's not just the model, right? The model is there, everybody can use it. It's all about how you leverage the model. And that is super complicated. And there it really performance increases lie in the detail. And I think our platform makes that part super easy. So I feel that we are very differentiated actually in that sense by really focusing on delivering the technology to the pharma companies.
So before we move to the last part, is there anything else you want to say about your company, potentially projects that you are at liberty to talk about, or is it something that you can't?
So, I mean, specific projects obviously can't do that. But one thing I would like to mention here is that we take an open source approach. This means that we bring parts of our platform on GitHub so everybody can use it and benefit from it. So I think it's something that anybody who is interested in exploring biofoundation models should check it out, because it's really made very beginner friendly. You can simply run very simple example notebooks, and we see many academics using it actually. So it's something I would encourage people to check out if you are interested in biofoundation models. And other than that, what I can say is that the use cases we work on with our customers, again, is all about how leveraging their data with existing models on use cases such as target identification, biomarker discovery, going from discovery actually to clinical stage use cases. So we are rather agnostic as a platform across this end-to-end chain.
Do you see more interest in the market for those approach lately? So is this accelerating? With all the noise around. I mean, noise is not a great term, but all the discussion around AI being used in pharma.
Yes, I mean, everybody is trying to find out what the right setup is, right? Should I develop as a pharma company my own huge foundation model that only I can use and nobody else? Is it leverage what's out there, build on top of it? Is it working with, I don't know, one company that pretends to have the best model on earth? So yeah, there's a lot happening in the market, and many explore different options. And we see that as well, and obviously get also, because of that market, a lot of opportunities to showcase how we can have and how we can solve their very specific use cases.
Okay, so looking ahead, what do you think is the most exciting about the role of AI in pharma at the moment and in the future as well?
Yeah, I think it's twofold. First of all, the sheer speed at which innovation is happening at the moment is really exciting. I mean, literally each week you go in and look into the latest papers, there's a new model released, a new step towards the virtual cell being achieved, a new benchmarking coming out. That's really exciting. I don't think that the field has ever seen that acceleration, and we are lagging a bit behind from the typical, let's say, traditional AI, text-based AI such as OpenAI. But things are accelerating, and that is amazing. Also in the research domain, not only in the pharma domain. And then, of course, the exciting thing for us is to see that we generate really good results now. I mean, we are capable of getting to very good things, which means that those models are working, and we're only at the beginning of those models. And if you then think about what the promise of this is, well, that's quite exciting, because the world is moving faster and faster, diseases as well. We saw what happened with COVID. So we cannot be in a position where we are stuck in a trial and error process that takes 10 years to bring something to market, right? It's all about speed of iteration. And the more you can do in silico, and I think that's what those models will help us doing, the faster we can be and the more impact we can have on bringing drugs to market faster. And I think it will happen. It is happening. So that's quite exciting.
How do you keep up with all the models, all the papers, and not only keep up, because it's great to know that it exists, but then also evaluate and see, okay, whether it's something you want to adopt or not?
Yeah, it's a very good question. To be transparent on a very old school way, but we have in our company, we have a Slack channel called Papers, and all our research are super connected to what's happening. And every time we see papers exciting, goes in there with a quick summary, obviously generated from a text model. And then the really exciting stuff, I think we started being very good at understanding what is really good and what is not. We decide to implement it, and then we move super fast. Evo 2 came out, took us, I think, 3 days to have it open source in Helical. And that was a very important step, and we consistently do that. So if there's something important, we deprioritize everything and we just bring it into the package, make it available to the community. And this is also how we build an authority with our community, right?
Yeah, actually, there is definitely a lot of value if you can do that quite quickly, because I know that in large pharma, there are definitely barriers in testing quickly the latest stuff, because most of the time there is discussion around security and all those things. So if you can solve this thing and evaluate things for the pharma community, then you definitely solve one of their issue, and that definitely has value. I mean, I've seen it in previous companies.
Yeah, 100% agree. And I think it's not that like people working in pharma are brilliant people. So it's not a thing of missing quality of that, but it's really organizationally speaking, very siloed. It's very, yeah, as you say, brought down because of regulation, and it makes sense. Again, you cannot take certain risks that maybe we can simply navigate a lot faster. So that is something we definitely add as value. Yeah.
I think that's what you said earlier. Pharma are not built for speed. And that's definitely, I think, something challenging them with all the super fast development in AI. It really forced them to rethink how they organize their tech department and how they evaluate everything. Because if you spend 6 months to evaluate a model, then there are already 5 new Chinese models that have been launched in the meantime.
Yeah, exactly. Fully agree.
Okay, last question. So if you had one piece of advice for a young entrepreneur who want to start a disruptive startup today, so what would it be?
One only. It's probably something many would say, but I think try to distinguish and be different is really important. Not for the sake of being different, but the new things are often the things that bring the disruption. So in our case, it was really taking like a model agnostic approach to this world, even though there were 2 models when we started. We simply believe that this is the shape the world is going to take. And by accepting this and not becoming a tech bio company that is using this model to build their own pipeline, I think we were very different and refreshingly different. And it brought us a lot of traction on the pharma side. So yeah, don't be afraid of, if you have like a bold, you think you have a good understanding of the market that nobody else has, then go for it and be different. Yeah.
Well, Rick, I think we'll finish on that. Thanks a lot. That was fun, and I learned a lot about foundation model. I hope people will learn a lot, and that will trigger their interest to deep dive into all the fun papers on foundation model, virtual cells, and I'm sure that will be more and more important, in the coming months and years. So definitely, you're working on in a very interesting area.
Thank you so much, Thibaut, for having me. It was a cool discussion.